Today we number in the billions, yet about 900 years ago, some genetic models suggest our ancestors fell by up to 99, a crash we unpack and why it matters. Many imagine numbers rising, but deep time holds booms and busts. You'll hear what DNA and sediments reveal, and where scientists still debate the bottleneck size and cause. What pushed them so far? When researchers take different genetic tools to the same deep time problem, they often find a similar shape to the past: a wobble or dip in inferred effective population size in the Middle Pleistocene. Earlier coalescent-based reconstructions such as PSMC and MSMC tend to show those dips as reductions in the effective number of breeding lineages, but they do not usually read as an almost complete wipeout. The recent, more dramatic claim, models that interpret the signal as a near extinction around 900,000 years ago, even suggesting a loss on the order of 99%, stands out mainly for its magnitude and its interpretation of how long and how uniformly that low point persisted. Part of the difference comes from what each method asks the data to do. PSMC and MSMC smooth information across genomes and across time in particular ways. Newer models fit alternative histories and can amplify short, sharp events or interpret complex structure as a single collapse. Where these approaches converge is important. Many of them agree there was less genetic connectivity and a lower effective population size deep in the Middle Pleistocene, reflecting a time when groups were more fragmented. Where they diverge matters more. One method may read fragmentation, migration pauses, or selective sweeps as a bottleneck, while another may treat those same patterns as persistent, spatially patchy populations, rather than a single catastrophic die-off. The truth may lie between these readings. The genetic signals point to a world of thinner connections and tighter refuges, but whether that means a few isolated groups holding on or a near extinction cleared to the genetic bones remains debated. Ahead lies the question of direct evidence. Can deeper genomic sampling, better calibration of clocks, or the rare biomolecules from old bones decide which picture feels truer? 900,000 years ago, the world felt less steady. Deep in the Middle Pleistocene, the rhythm of ice and warmth returned, but the beats changed. Where earlier swings had been more regular, the climate began to push into longer, harsher episodes. Glaciers grew, larger seasons sharpened, and the margins where wet met dry moved more often and more violently than before. Evidence suggests these changes did not unfold as a single catastrophe, but as a prolonged tightening of realities for the creatures who read their lives from water, plants, and prey. On the ground, this meant landscapes that could flick from plenty to scarcity within a lifetime. Lake shores that once held reeds and fish pulled back to cracked mud. River corridors, thin to a trickled dust, rose in wind over wide plains. Forest patches frayed into savanna. Trees retreated to scattered hollows. For hominin groups, cooking fires would have been smaller, food stores thinner, and seasonal rounds more uncertain. The ache of hunger and the labor of travel were real. Bones show where stone tools show repair, and sites suggest people moved to the places that still held water and edible plants. These shifting geographies and seasons are a plausible, even patient stress on populations. Reduced habitats and punctuated isolation could cut connections between groups, lowering the effective sizes that genomes later read as contractions. That pattern, many small refuges separated by expanding deserts and ice, can look in genetic models like a dramatic crash, even if a larger number of people lingered in fragments. The story is not settled. The record asks for care. In the next passage, we will turn to the layered marks in ocean and ice, the long archives that help time these changes, and see how they frame the cool interval around 900,000 years ago. Stone tools last. A well-made hand axe is an archive. You can hold the rhythm of the strikes that shaped its edge, the faint polish where skin slipped on hafting, the warmth of a stone recently used to break bone. For long stretches of the past, this kind of core technology was remarkably stable. Archaeologists argue that the Acheulean package, the hand axes and cleavers and the know-how to make them, was kept alive and taught across generations, in some places for well over 1 million years. That continuity acted like a cultural insurance policy. When landscapes shifted, groups could carry a reliable toolkit and the skills to use it into new settings. Stones speak a common language across rivers and valleys. The shape of an edge could be read and reproduced by someone far from where it first appeared. But durable tools are not a demographic miracle. Genetic evidence shows low diversity in later hominins, a pattern many interpret as the trace of past contractions in population. Size and models suggest that cultural links between groups could themselves lower the effective size of populations. So even while Acheulean methods smoothed daily risks, giving hunters and gatherers practical ways to access meat, marrow, and plants, they did not automatically prevent deep stresses. If climate pushed habitats apart, if water and game retreated into scattered refuges, technology alone could not knit isolated bands back together. How then should we read a long-lived toolkit as proof of unstoppable resilience, or as a fragile thread that is sometimes held and sometimes frayed? The answer lies in the lives between stones, the movements, the exchanges, the learned hands, which we will explore next as we consider the networks and knowledge that kept people going. Tonight, imagine the sweep from our crowded planet back into a thin, uncertain moment deep in the Middle Pleistocene. Billions now, a whisper then. Some genetic models point to a dramatic reduction in our ancestors centered near 900,000 years ago. Indeed, one line of work places a sharp contraction between 930 and 813 years ago. Read one way, those models suggest the breeding pool fell to only a few thousand or perhaps even a few hundred people. Other readings propose less extreme reductions or later, milder ones that leave several thousand adults surviving. These are not settled facts but interpretations of patterns written in DNA. The same patterns can mean different things: a true near collapse, a long period of fragmented groups, or shifts in how lineages mixed. Picture a low, pale sun on a cracked lake bed, the wind carrying dust where camps once clustered. An evocative scene, but one the hard record must prove. Remember that genomes mainly record those who reproduced and passed genes, not every person who lived and traveled their lands. In the next sections, we will walk that thin line between possibility and proof. We will listen to genomes that carry shadows of the past to the scarce bones, and the Acheulean tools that endured into marine and lake sediments that record pulses of drought and dust. We will lay out how climate could have carved Africa into scattered refuges, how social networks and simple technologies might have buffered hardship, and how choices in analysis change the story we read from DNA. Dramatic headlines that suggest entire peoples were wiped out overstate model-based claims. Instead, we will calmly unpack the evidence and the debates weighing what is well supported and what remains provisional. Shall we begin? We will travel this story together by holding 4 kinds of evidence side by side and letting them illuminate one another. First, the patterns written in genomes, statistical models, and ancient DNA give us a long branching picture of ancestry and change. Some models point to a deep contraction in our African ancestors between 930 and 813 years ago. Others read that same genetic signal as the result of population structure or shifting connections between groups. Second, the bones and the stones speak in a different key. Fossils are rare and often hard to classify, and toolkits and site records can show both continuity and gaps. Third, the world's slow archives, sediments, faunas, and climate records, record rhythms of wetter and drier. These more connected and more fractured landscapes would have shaped where people could live. Finally, ecology and behavior, diets, group networks, and mobility offer plausible mechanisms for how populations rose and fell across that shifting stage. Put plainly, genomes preserve echoes of births, deaths, and movements across generations like rings in a tree. Picture lakeshores and scattered hearths where small bands stretched thin across the landscape. At each step, I will mark what is well supported and what remains provisional. Genomes can point to tight bottlenecks or to low effective sizes caused by social structure. Bones can be absent from a region without proving it empty. Climate archives can show stress without assigning single causes. We will watch where different lines agree and where they contradict. We will listen for the difference between a true near extinction and a portrait of many small, semi-isolated groups. And then, with that cautious map before us, we will ask the next practical question: what does 99% really mean? When geneticists talk about a collapse of 99, they are usually talking about effective population size, Ni, not a simple head count. Effective population size is in essence the size of the breeding pool. With mitochondrial DNA, for example, that pool is the line of mothers whose genes are passed down other parts of the genome sample, other subsets of breeders. The number that comes from these genetic calculations can therefore be much smaller than the total number of people living on the landscape. That is why different researchers have proposed very different portraits. Some readings of the data suggest our ancestors may have been reduced to a few thousand, or perhaps even a few hundred breeding individuals. Other mainstream estimates place surviving adults in the tens of thousands. These are model-dependent pictures, and they sit in active debate. Put plainly, Ni tracks the effective number of genetic contributors across generations, not every living person at a single moment. A modeled drop in Ni can mean several different things. It can mean a true sharp reduction in the number of breeders. It can also mean long-lasting population fragmentation, where many small groups live apart and exchange few mates, making the genetic pool look smaller. It can reflect changing patterns of movement and interbreeding across regions, or simply the way variation was shuffled before later expansion. Small breeding numbers raise risks. Chance events can wipe out tiny groups, but a low Ni does not always demand a one-time global near extinction. Picture small groups around a drying lake margin, their paths and meetings growing rarer, and their genetic exchanges thinned. To understand which story the genes tell, we must next listen to the slow clock of mutations and generations, and see how genetic time is measured. The genome keeps time in tiny scattered marks. Mutations, the small changes that pass from parent to child, accumulate like scratches on bone. If we can count those scratches and know how fast they appear, we can turn genetic difference into a measure of time. That is the basic idea behind the molecular clock. But two parts of that mechanism are uncertain. One is the rate at which mutations occur. Many researchers treat it as roughly constant, yet whether it has been perfectly steady through deep time remains an open question. The other is generation time, the average age when people had children. Without a good estimate for that, converting genetic change into years is imprecise. Mitochondrial DNA, which mutates faster than the rest of the genome, has been especially useful for closer time frames because its pace reveals more recent twists in the story. Put simply, a greater number of genetic differences usually implies a longer separation in time between lineages. This is why careful calibration of the molecular clock matters for every inferred demographic event. How can a handful of mutations be turned into a clock? The clock is usually set by tying genetic differences to dates we trust from the ground, fossils, or clear geological events, and checking that the ticks match. Change either the tick rate or the assumed generation span, and the whole timeline slides. Events move earlier or later, and a gentle dip in inferred population size can deepen into what looks like a dramatic collapse. In short, the gears of demographic inference, mutation counts, mutation rate, and the calendar of generations determine both the timing and the scale of ancient population events. Next, we will trace those shifting signals backward through lineages to see how histories are read from living genomes. Imagine each genome as a long family album, its pages filled with tiny marks left by mutations and reshaped by the slow shuffling of chromosomes. Coalescent thinking is the gentle backward sweep through that album. Instead of watching families grow forward in time, we trace today's genetic lines back until they meet at common ancestors. Those meeting points carry information about how many ancestors there were and how often different groups mixed. Recombination, the random reshuffling that happens when eggs and sperm form, breaks the genome into many independent threads, and mutations pin small dated marks along those threads. By reading the pattern of shared and unique marks, researchers infer whether many lineages stretched far back or whether most lines trace to a few recent ancestors. How do we read that book without seeing the past directly? We treat mutations as dated marginal notes, and recombination as the book's worn spine, so patterns of marks reveal when lines converged and diverged. We do it by using computational approaches that summarize different features of genetic variation. Some look at the time to common ancestry along long stretches of DNA. Others combine many genomes to capture subtler shifts. Still others use the distribution of how common or rare particular variants are across people. All of these methods turn mutation and recombination into clocks, and all are sensitive to the clock's settings and to the way populations were structured or connected. Cultural barriers, isolation, and selective forces can leave patterns that mimic shrinking numbers, so results must be weighed against archaeology, climate, and fossil clues. Next, we will meet a recent study that used one such approach to propose a dramatic bottleneck deep in the Middle Pleistocene, and consider what that claim means for the larger story. In 2023, a team turned the living record of human DNA back on itself. Using 300 modern human genomes and a new statistical model, they projected variation far into the Middle Pleistocene. Their reconstruction points to a severe and prolonged reduction in the ancestral population, roughly between 900 and 800,000 years ago. The headline that followed framed this as an almost complete wiping out of diversity across many lineages. A very small ancestral pool holding on for an extended span. The authors even suggested this interval could have shaped later branching in our family tree. That is the plain claim. The genetic signal looks like a long, deep narrowing at a time when the world was changing. Put plainly, the signal reflects how many ancestors contributed genes forward, not a headcount of every person alive then. Like sampling water downstream, the pattern can speak of droughts upstream without showing every side stream. Yet this is one way to read patterns in modern genomes, not an uncontested verdict. Other researchers note that structured populations, culturally channeled migration or shifts in how groups exchange genes can produce similar signatures. Commentaries soon appeared urging caution and deeper testing. The modern samples are powerful but indirect, and the farther back we look, the more models and assumptions steer the outcome. We lack direct ancient DNA from that deep time in Africa, so the story rests on how we translate present-day variation into past demography. Imagine lakeshores thinning and river corridors narrowing, with groups becoming more isolated. Such landscape changes could create genetic signals that mimic a collapse. How small that ancestral pool really was, and what caused the change, will be clearer when we examine what these numbers mean next. When you first hear that our ancestors may have been reduced to a very small number of breeders deep in the Middle Pleistocene, it feels like a clean, sharp fact. In truth, those small figures live inside models. Genetic data do not speak in plain numbers, they speak in patterns that researchers translate with assumptions about mutation rates, generation time, and the way populations mixed or stayed apart. Different models can take the same pattern and produce quite different narratives. Some work points to a severe contraction in the interval roughly between 930 and 813 years ago, but how severe depends on choices made before the math begins. A headline number is a single point on a wide landscape of uncertainty, confidence intervals, alternative model forms, and the samples used all shape that point as much as the underlying DNA does. That sensitivity matters because a model dip in the pool of breeding ancestors can mean several things: a true near-global collapse in headcount, a long period of fragmented, partly isolated groups whose genes drift apart, or shifts in how selection acted on certain parts of the genome. Each of these scenarios leaves overlapping traces in modern genomes, and each can be emphasized or muted by different analytical choices. So when a study reports a very small ancestral breeding population, treat that as one plausible reading of a tricky signal, not a single settled truth. How we parse that signal depends on how we model the past, how we measure our uncertainty, and how broadly we sample living variation. Think of models as maps, not the territory, helpful but imperfect guides. Next, we will look closely at how population structure, natural selection, and limited sampling can change the story these numbers tell. When genetics whisper of a dramatic loss around 900,000 years ago, several caution flags wave in reply. One is population structure. The record and models both suggest our ancestors were often spread across shifting habitats, pockets of groups along rivers, lakes, and coasts, sometimes connected and sometimes isolated by climate. That patchwork can make a long divided population look like a sudden collapse when we read it through genetic summaries. A second caution is natural selection. Selection can trim variation in parts of the genome, and over long stretches this trimming may change the patterns we use to infer past sizes. In plain terms, forces that shape bodies and behaviors can also shape the genetic footprints we try to interpret, so the same signal might mean ecological stress, selective change, or both. Put simply, effective population size measures how much genetic diversity is passed to future generations, not the raw headcount at a camp. Imagine small bands camped along a shrinking river, their fires low in lean years and bright in plenty. Those living rhythms cast genetic shadows we must read carefully. A third and equally important caution is sampling. Fossils and genetic surveys are sparse and uneven, especially across Africa, and that patchiness can bias conclusions. If we lack data from key regions or lineages, models may overstate how small ancestral populations were, or how sudden the change appears. Many researchers therefore treat a 99% reduction as one possible reading of the data, not a settled fact. The debate is active. Some see a true demographic squeeze, others see structure selection or gaps in sampling playing large roles. Testing these possibilities means comparing models, expanding regional genetic sampling, and seeking archaeological or environmental signals that match or contradict the genomic story. How do other genomic approaches and the rest of the evidence agree or disagree with these warnings? Genomes feel like time capsules, but most of them are fragile. The recoverable span of ancient DNA in the record reaches back only tens of thousands of years in many cases. The woolly mammoth is an example of a species for which we can reach a little farther. That means the era deep in the Middle Pleistocene is largely out of direct reach for ancient human DNA. Neanderthal genomes, however, do give us a clearer clock on parts of the tree. Comparisons of Neanderthal and modern human sequences have been used to estimate the point at which their lines last shared DNA, a time often placed deep in the past. Those sequences light up branching patterns and help calibrate genetic clocks, but they come from populations outside Africa, and therefore cannot by themselves tell us what happened to humans living across the African continent at the same deep times. They act like a lantern on one path, rather than a map of the whole landscape. Genetic models from modern genomes do suggest dramatic scenarios. Some work has proposed very small ancestral breeding numbers around 1 million years ago, but those inferences are model-dependent and indirect. Neanderthal genomes illuminate divergence and some demographic detail in Europe and Asia, yet they do not directly sample the African populations, whose structure, connectivity, and local extinctions would shape any continent-wide bottleneck signal. In short, the deepest episodes of African demographic history remain a puzzle, partly because the molecule we most often read, ancient DNA, rarely survives that long. The next windows into that past come from different molecules and the fossils themselves. Ancient proteins preserved in tooth enamel can survive far longer than DNA and help place fossils on the family tree. It is a promising but limited window. When DNA has long vanished from a fossil, some molecules quietly remain to tell part of the story. Proteins locked inside mineralized tissues can survive where genetic material will not. Their chemistry carries echoes of ancestry, small cumulative changes that researchers can read to place a fossil on the family tree when DNA is gone. Early comparisons of proteins once helped reshape ideas about relationships among apes and humans, and more recently the same logic has been applied to deep human fossils. The work feels gentle and exact. A tooth or a fragment of bone as a tiny archive, its molecules slow to fade. Tooth enamel and dense bone trap proteins within mineral layers, limiting bacterial attack and weathering, so chemical signatures can persist far longer than DNA. Think of the molecules as ink sealed under varnish, holding a faint script of kinship through time. This approach does not give the whole picture. Proteins are good at refining who is closer to whom, helping to sort early Homo fragments into more secure branches. They do not, however, carry a clear census of past people. In plain terms, protein sequences report ancestry at a handful of preserved molecular sites, rather than across whole genomes, so they sharpen relationships more than they estimate past population size. Where genome-wide data can suggest past population sizes, proteins usually cannot tell whether a low genetic signal came from a single catastrophic die-off or from long-standing population structure and limited gene flow. In other words, proteins help with relationships. They give only modest demographic detail. Listen to the image, a worn tooth, its pale surface glinting under a lamp, holding a faint molecular memory of long-ago kinship. In the quiet lab, a gloved hand turns the specimen, the enamel cool, and ridged from a lifetime of chewing, while instruments read molecules that remember lineage. As those molecular clues sharpen our map of who was related to whom, we turn next to the stage on which they lived. The shifting climate and landscapes that might have pressed populations into narrow places. What did the world feel like then? We will listen to the planet's longer records, ice sediments and seas, that carry the rhythms shaping habitats and human chances. If you want a single clock that marks long swings of cold and warmth, scientists often turn to the ocean. Tiny differences in oxygen atoms preserved in seabed cores, ratios of heavier and lighter oxygen in shells, record the planet's temperature. Put simply, when more water is tied up on land, as ice seawater becomes richer in the heavier form of oxygen, and tiny shelled creatures lock that signal into their skeletons. Those patterns are the basis of the marine isotope, stages a numbered rhythm that sorts cooler, even-numbered stages from warmer, odd-numbered ones. The isotope record extends across the past 900,000 years and beyond, giving researchers a long, repeating beat to read. From this long file of shells and mud, paleoclimatologists have mapped more than 100 such stages since the Pleistocene, and they use the latest one, a warm interval, as our present reference point. Around 900,000 years ago, the global record shows a change in tone. Marine cores, dust layers, pollen sequences, and cave flowstone together indicate stronger cold spells and increased signs of glacial influence. Heavier oxygen in shells, thicker dust, deposits blown from exposed plains, and shifts in vegetation preserved on lake beds. These proxies point to broader, harsher swings in climate that would have tightened habitats on land. Lakes shrinking, forests retracting, grasslands shifting. Across those landscapes, shorelines could have left bands of cracked mud and wind-blown silt, and rivers became lifelines squeezed between thinning woodlands. It was as if the climate dial had been turned toward extremes. The picture is not of a single catastrophe, but of a world where the weather's mood grew louder and more extreme. How those louder beats reshaped where people could live leads us next to the fragile story of African hydroclimate on a knife edge. Around 900,000 years ago, between the interval some researchers mark as a sharp contraction, Africa's water changed its mind often. Deep-sea cores and the long memory of river and lake beds closer to shore suggest rainfall came and went in pulses. Monsoon rains and large-scale shifts in ocean atmosphere patterns played a role in deciding whether a basin brimmed with life or cracked to mud. In the places where people lived, a green ribbon of gallery forest could retract to a thin line along a river, then return a few generations later. Lakes and rivers that had once fed many mouths fell to low levels or dried seasonally, and the map of usable land broke into islands of habitat. These changes could unfold within a few generations, repeatedly rewriting routes to food and shelter. Foragers moved within that shifting mosaic, patches of forest, open grassland, and expanding dry country rearranging across the landscape. Food and water became unevenly distributed, mobility grew more costly, and groups that once exchanged mates and knowledge could find themselves farther apart. Stone tools and careful knowledge of plants and animals helped, but the rhythms of rain and drought could still strain even skilled hunters and gatherers. Sometimes a careful knowledge of water holes and dried plant stores bought time and linked families across distances. As refuges shrank and scattered, the web of social ties that carried genes and culture risked thinning. Imagine standing on a lakeshore that vanishes over a single human lifetime, shorelines gone, trees, retreating corridors cutting off. Could such repeated pulses of aridity and fragmentation create the genetic signature some models read as a drastic bottleneck? The next section follows those shrinking ribbons of water and land, river corridors, lake margins, and coasts, to see how scattered refuges might turn a continent into a maze. The world I want you to feel is a patchwork of green and bare earth, a map that kept changing with the weather. Where rivers ran, life clung to the wet margins, where lakes held water grasses and fruiting plants clustered along coasts, tides and rocky shores offered another steady, if shifting, supply. As climate swung, those places widened and then thinned. Whole corridors of movement could close like a gate, leaving pools of people stranded in the most reliable patches. Some researchers note a severe contraction of our Africa-based ancestors between 930 and 813 years ago. Even without insisting on a single cause, this image of shrinking refuges helps explain why populations might have become scattered and isolated. Genetic patterns we read today often carry the echoes of long isolation, where rare variants vanish and others rise by chance. Like islands in an archipelago, these refuges could separate groups and slow the flow of genes and ideas. Picture a shrinking shore where only a few families return each season, carrying a narrow set of skills and local memory. When groups become thinly spread across river valleys, lake edges, and coastal strands, their connections fray. Bands that once exchanged mates and ideas find themselves farther apart. That isolation can leave the genetic marks of a small ancestral pool, even if many people lived in separate pockets. The archaeological record suggests long stretches of hard living, fewer sites, more episodic occupation, and some scholars argue this could amplify genetic drift and loss of diversity. In material terms, this often looks like brief, repeated visits to the same reliable place, rather than steady settlement. Those who kept watch at those lake margins, passing knowledge of where water still ran, and which plants to trust, became cultural anchors for others. The next section looks outward toward the hominins who might have filled those narrow places. Across Africa, Eurasia, and the islands, beyond, a small family of human-like creatures traced their lives across shifting rivers, lake shores, and open plains. In many accounts, the long-lived H. erectus group, including local varieties often called H. ergaster, dominates the picture, and was the first to push beyond Africa. At the same time, other more ambiguous, archaic forms were emerging. Classifying those fossils is hard. Scholars debate whether differences mark distinct species or regional variety. East Asia is one region where many fossils are routinely placed with H. erectus, while parts of Europe yield forms that frustrate neat labels. The hills at Atapuerca remind us how change can be local and sudden. One set of people there showed a mix of erectus-like features and traits later seen in modern humans, yet they left no clear continuity with the people who followed. The record is sparse and episodic, a scatter of bones and stone in a landscape that could turn on a season. Think of these fossils as dialects of a long conversation, not always separate languages. From the flooded coasts of island chains to the cooler river valleys of Europe, these hominins lived with the weather's moods and with each other's differences. Populations rose and fell. Extinction was the likely fate of many Middle Pleistocene groups. Some regions show long persistence of a recognizable erectus lineage, others a patchwork of arrivals and losses. Such fragmentation, pockets of people living, dying, and sometimes meeting, makes the past look complex and unsettled rather than cleanly split. Stone tools and the traces of daily life still survive. The next question is what those tools were and how they carried people through a world in flux. Imagine small groups along lake edges shaping stone, while water marked the seasons. In many places across the Old World, people carried a familiar set of stones bifaces. We call hand axes broad-edged cleavers and simpler core and flake pieces. Evidence shows these forms were widely used and surprisingly stable through long stretches of time. Hand axes hardly changed shape through a million years, yet later they show a slow trend toward thinner, more refined examples. Some regions are marked by particular notes. Some sites have many cleavers or very large axes that may have been made for display or heavy tasks. Other places show Alduin-like flakes and cores continuing alongside the larger tools. Names like Jesher Benat Yaakov Alorgesili and Isamila appear in the record as places where these toolkits were made and used. The Indian subcontinent also preserves Acheulean artifacts, along with a partial cranium that hints at the people who made them. Their persistence through time hints at shared learning and repeated practice, passed down where groups lived and moved. Those stones speak of adaptability as much as sameness. Across different raw materials and prey, toolkits shift in frequency and form. In some places, fine, thin hand axes appear where good stone could be thinned. In others, heavier tools appear where large carcasses or different tasks demanded weight. Simple flakes served quick cutting or scraping. Some tools feel like workmates by light of dawn. A flake biting hide a cleaver parting bone, the dull thud of marrow cracked open. A hand axe could sit like a trusted knife at a camp, its edges retouched as rhythms of use and repair demanded. The Acheulean is both a long, steady tradition and a flexible set of solutions to varied landscapes and meals. How these implements met the shifting menu of survival, hunting, scavenging, coastline foraging, is the next quiet question we follow. When archaeologists turn over those ancient surfaces, the picture that comes into focus is not a single strategy, but a patchwork of ways to eat and survive. Many researchers now say the old argument, were our ancestors hunters or scavengers, is largely a false choice. Intelligence and opportunity made both possible. In some seasons and places, people favored small, safer prey, or scavenged. In others, they set to the dangerous work of skinning and disarticulating large animals. A famous landscape shows hand axes used to butcher horse, deer, and even rhinoceros, with bones bearing marks that suggest people were intent on getting every last bit of nutrition. At the same time, the bones that reach us are biased by the earth itself. Taphonomy reminds us that some assemblages over or underrepresent what people actually ate. Put plainly, soil, water, and time erase much of the menu. What survives is a biased sample. Bones chewed by carnivores, tools scattered by weather and deep burial all shape what we find. Archaeologists read those filters with caution. Plant foods and coastal resources likely filled gaps when meat was scarce, and the ability to process or store resources mattered for survival, as later debates about food storage show. Seasonal rhythms of plants migrating herds and shorelines would have shaped a plural diet. Marrow and rich tissues when available, small to medium game, and gathered plants at other times, opportunistic foraging, where coasts or rivers offered shells and fish. This diversity made life flexible, but also tied populations to shifting landscapes and seasons. Imagine a shoreline at dawn, salt in the air, hands prizing open shells, and quiet talk as protein is gathered. Like a seam between seasons, these resources stitched together a changing diet. Fire, when regular, could add another stitch to that seam and give communities a steadier reserve in cold months. The next section looks more closely at what the sparse fire record actually shows. Fire in the deep past comes to us in small local traces rather than a bright continuous line. A few sites show convincing burning, caves in southern Africa dated near the Middle Pleistocene, and the lakeside camp at Jesher Benat Yaakov where tiny burnt flints mark what look like ghost hearths. Other places, Britain later in time, show clearer hearths, and within the last part of the Pleistocene, many caves accumulate burns in repeated layers. Yet the majority of early sites simply lack signs of fire. Caves themselves do not always keep a long memory. Soft rock shelters erode and vanish on timescales of a few 100,000 years, and the frequency of preserved hearths can fall for reasons that have nothing to do with people's choices. So the picture that survives is patchwork flickers of human-controlled fire here, and they're not a continent-wide everyday glow. A single hearth would have warmed a handful of people, its light fragile against the night. Like a candle in a dark hall, these hearths are intimate local signals rather than broadcast beacons. That patchiness matters for thinking about a demographic pinch near 900,000 years ago. If habitual reliable fire use were already universal, it could have softened cold or dry swings by improving warmth, food safety, and nighttime defense. But the record suggests otherwise. Burning was local and episodic. The techniques for making and carrying fire were imperfectly preserved in the archaeological record, and even where hearths exist, their food remains do not always show clear cooking. Could fire alone have saved small, scattered groups? The evidence indicates it was unlikely to have been a global buffer at that time. Night predators and thin networks of people still shaped survival, threads we will follow next as we listen for the sounds of the night watch. On a cool wind-breath night, the world of early Homo was full of other mouths and sharp teeth. Large carnivores patrolled the edges of water and plain scavengers, and omnivores moved in the same places where people followed herds. The fossil and isotope evidence we do have reminds us that competition mattered deeply. Even species that were not strict meat-eaters left traces of stress in the larger predator guild. In that long Middle Pleistocene interval, some genetic work points to a severe contraction of our Africa-based ancestors between 930 and 813 years ago. Whether that contraction was a single brief cliff or a drawn-out tightening, the everyday reality for people would still have been the same: keeping a fresh carcass away from prowlers, sharing meat before it's spoiled, and watching the dark together were as necessary as any tool. Such genetic contraction could mean fewer people, or simply long isolation between groups, leaving daily dangers unchanged. On nights like that, blood and embers scented the air, while a sentry's shadow moved along the dark. Archaeologists and geneticists piece these traces together to see how people coped. Stones can cut and throw, but people working together change the odds. Vigilant sentries, noisy fires when possible, simple tools held like small promises. These are social technologies. They let groups defend kills, move wary children and elders to safe places, and pass practical routines from hand to hand. Later debates about speech and complex symbols touch on the same theme. More effective communication makes collective defense and provisioning easier. Yet even without elaborate material, culture cooperation could be the difference between survival and fade. What did that cooperative life look like on the marginal edges of habitability, in places that would later act as fragile frontiers? The story of Europe in the deep Middle Pleistocene is one of edges and echoes. As ice and seasons swung wider and colder around the 900,000-year mark, much of the continent became a marginal place for people shaped for warmer mosaics. The fossil and stone record there is thin and patchy fragments from sites like Boxgrove, Pakefield, Swanscombe, Arago, and Petrolona appear and disappear through the layers. In some places, A scatter of flakes and bone shows a brief presence in others, a richer horizon hints at a longer stay. Pakefield has been read to imply much earlier visits in some estimates, and by the midpoint of the Middle Pleistocene, a more substantial human presence is recorded at certain English sites. But classification is difficult, and the sequence is irregular. Wine dust and cold could erase camps a hunted carcass, and a hand axe may be all that survives to speak for whole seasons of living. Bones break, tools move downslope or are trampled and sediments shift, so a few artifacts can stand in for many lost lives. Picture a riverbank where a hand axe damp with use catches a thin winter light while the wind carries away footprints. These traces suggest boom-and-bust settlement rather than steady growth. Small groups pushed into river valleys, lake shores, and sheltered lowlands. When climate opened a window, they retreated or vanished when conditions turned harsh. Archaeology most often preserves the successes, those camps and toolkits that left durable remains, while failures fade. Booms were brief pulses of occupation, busts could be long, empty centuries. Scholars describe episodic expansion and local extinction as a likely pattern, a slow probing of a frontier that was at once inviting and unforgiving. Like footprints in shifting sand, the archaeological record captures only moments of passage. What routes did those pulses follow, and which gateways let people into and out of the European margins? Tracking those paths brings us to the corridors and coastal gates that shaped ancient movement. The Levant and the Strait at Bab al-Mandeb were not fixed doors but shifting thresholds. In the Near East, the record shows repeated comings and goings. Early modern people and other hominins appear, then vanish as climates turned dry and resources dwindled. Eskul and Kafse are quiet reminders that an advance could end in retreat when the rains failed. These openings were not brief flickers, but windows that mattered across many generations. Along the southern gate, lower seas and friendlier rains opened a coastal route out of eastern Africa into Arabia, and beyond a corridor that later people used to reach distant shores, and by another interval crossed by boat to Australia. Like lanes on a highway that narrow with traffic routes widened or constricted with climate. These passages opened and closed with tides of water and sky. When corridors were open, groups mixed. When rain failed or sea rose, those links frayed. Imagine river margins drying to cracked mud, a salt breeze shading a bare beach or a lake that once fed many mouths reduced to a string of ponds. Populations pushed into riverine refuges and coastal pockets isolated by deserts or high seas. Genetic methods pool signals over space and time, so reduced exchange among pockets can mimic demographic collapse. Evidence suggests such intermittent connectivity can fragment populations into semi-closed networks. In a dried river hollow, a few family groups might share food and mates for generations, shaping who survives. That pattern of many small, partly isolated groups can look in genomes like a sudden collapse of numbers, even if total headcount remained distributed across refuges. Understanding this distinction helps us read genetic patterns more carefully. What appears as a sharp bottleneck in one model may instead be the echo of doorways that opened and shut across the map, and those shifting gates shape the tangled web of lineages we begin to untangle next. Deep in the Middle Pleistocene, as climates swung and landscapes reformed, the family tree of our deep ancestors was not a neat fork, but a braided web. Many lineages lived, and then vanished. The record hints that these interactions stretched on for nearly 2 million years. Some genetic work points to a period when the long-term effective population of our African ancestors may have been only on the order of 10,000 breeding individuals, and when the effective female population could have been under 5,000. Those are measures of genetic diversity, not literal headcounts. They arrive wrapped in assumptions about generation time and ancestral variation, and those assumptions push and pull the inferred timing of splits and losses. Seasonal moves and local disappearances could thin genetic signals only to swell again when groups reconnected seasonally. Imagine the clack of stone on stone, a riverbank at dusk, the low hum of groups moving between favored campsites. Life continued, but its genetic trace tightened and loosened with each season. That braided pattern makes demographic reading difficult. If the ancestral population already held a lot of variation, a split between the lineage leading to us, and the branch that produced Neanderthals and Denisovans can appear recent or more ancient depending on which fragments of the genome you study and what rates you assume. Occasional exchanges of genes between groups or the repeated disappearance of local lines will blur signals in the same way a handful of small, isolated groups can mimic the genetic footprint of a single severe crash. Might a web of small groups exchanging genes now and then look in the genome like a catastrophic bottleneck? The next section considers that alternative how structure and patchy connections can masquerade as collapse. Imagine a patchwork of small bands living like beads on a broken string, some near lakes and river margins, others clinging to coastal shelves or patches of greenery. When climate pressed in those patches shrank, groups became thinner and more isolated. They still met, traded, and sometimes mated, but contact was intermittent. In that kind of metapopulation, the total number of people across the landscape can be much larger than the number of effective breeders whose genes flow freely. Modeling by some researchers shows that culturally guided movement between such subdivisions can suppress effective population size. That means the genetic signal looks like a sharp decline, even when many people persisted in scattered refuges. Put plainly, effective population size measures how many people actually contribute genes across generations, not the headcount living on the landscape. When movement and marriage links weaken, the genetic estimate falls faster than the true number of people. Isolation, local die-offs, and occasional reconnection can wipe out local lineages and speed the loss of genetic diversity. Random changes have more power in small, separated groups. Surviving communities pass on only a slice of the old variation. Over centuries, the genome records this as a narrow gate in history, similar to what a single catastrophic crash would leave behind. Think of many small libraries across villages. If only a few books ever circulate, the shared catalog will look thin even though buildings remain full. On a cool dawn beside a shrinking lake, smoke from a single hearth can mean life continues even while genetic traces fade. The point is simple, and subtle structure can mimic collapse. So when we read a deep dip in genetic reconstructions, is it the footprint of near extinction or the echo of many small shuttered neighborhoods slowly reopening? How would a true near extinction look in DNA? If almost an entire ancestral population had truly vanished, the trace in our genomes would be simple and stark. Many chromosomes would look more alike than you'd expect, with far fewer places where the two copies differ. A deep genome-wide loss of heterozygosity. Large stretches of DNA would be identical by descent, long shared tracks inherited from the few survivors showing up across descendant groups wherever their lineages later spread. Such patterns would not be a local quirk. They would be visible in genomes from different regions, like a single narrow doorway that every family once passed through. That signal would also leave fingerprints in mitochondrial lines, making overall mitochondrial variation unusually small compared with what we see in some living primate populations. The actual picture from research is more mixed. Some measures of human mitochondrial variation are indeed low, low enough that some have argued our ancestors once dropped to only a few thousand or perhaps even a few hundred breeding individuals. Other analyses suggest larger ancestral breeding numbers closer to 20,000 in deep time. Modern genomes also carry small amounts of other hominin ancestry, a reminder that mixing and survival can be complicated. A few percent of some genomes today traces to different branches. Population structure sparse sampling, and the patchy fossil record can make a modestly fragmented past look in genetic terms like a sudden collapse. Survivors, if there were few, would cluster where water and food lingered, river margins, lake shores, or productive coasts, places that buffered risk and kept stories alive. Stone keeps its own memory. These material traces help us read where people lived and moved, even when genomes alone leave questions. Next, let us turn to the bones and stones and ask what the fossil and artifact record adds to this story. The bones and stones we find are fragments of a much larger story, and they arrive to us after a long and uncertain journey. Paleontologists have long warned that the geological record is imperfect. Darwin spoke of preservation, and sampling bias, and later thinkers suggested that species sometimes appear and disappear in fits, quick pulses that leave gaps. Between about 1 million and 350 years ago, researchers struggled to classify many fossils precisely, while East Asia stands out as a region where most specimens are consistently placed with a particular form. In the field, you can see why a scatter of teeth on a dusty slope, a stone tool half buried in wind-blown sand, whole seasons of people simply not preserved. That patchiness means absence of evidence is not evidence of absence. Does a gap mean a population collapsed, or that the right place was not sampled, or that soft things never fossilized? A helpful image is a book with many torn pages. Fragments hint at the plot, but we must not read a single missing chapter as ruin. At the same time, the fragmentary record does not by itself demand a dramatic near-total wipeout. Sudden changes in who appears where can come from local replacements, shifting habitats, or taxonomic uncertainty, rather than a single catastrophic die-off. Stone tools and sporadic bones tell us about presence and behavior, but they are poor gauges of continent-wide headcounts. To move from an empty layer to a claim that most ancestors vanished requires multiple lines of evidence agreeing: the genetics, the ecology, the sediments. For now, the bones and artifacts ask us to be cautious and curious. Next, we will listen to the earth itself, reading climate and landscapes for the pressures that might have shaped those gaps. Dust, bone, and footprints remember shifts the eye can miss. Across the African plains and woodlands, the fossil record and animal genetics tell a quieter story of change. Many large mammals reorganized their ranges and fortunes as climates swung and habitats broke into smaller pieces. Some scholars have argued that these long, repeated cycles of fragmentation could drive populations to isolate, adapt locally, or disappear altogether. And a notable contraction of our Africa-based ancestors is often placed in the interval between 930 and 813 years ago. Ancient DNA from long-dead animals has already begun to add depth to these faunal histories, showing that species did not respond uniformly. Some tracked shrinking habitats, others vanished from places they once filled. The landscape of that time was not a single uniform wilderness, but a shifting patchwork where food, water, and shelter moved like a slow tide. For people making a living in that patchwork, the consequences would be immediate and practical. Species that tend to stick to familiar environments are pushed to follow those environments or face extinction. hominin groups, would likely do much the same, moving with prey clustering in favorable valleys and losing connections when corridors closed. That process, many small groups pulled apart by changing ecosystems, can leave the same genetic footprints as a dramatic die-off, even if headcounts never fell to near zero. In plain terms, fragmentation cuts off movement and gene flow, so small groups drift genetically like islands in a sea of grass. You can imagine waterholes as temporary meeting places, dust rising under herds and hunters alike. These patterns echo in other primates. As we turn to compare these patterns with the lives of other primates, we can ask gently, what do our cousins' fluctuations reveal about resilience and risk? When trees thinned and rivers narrowed, humans were not the only apes to feel the squeeze. The great apes of Africa responded to the same shifting map of rain and drought in different ways. Gorillas range into higher, cooler forests and can rely on abundant leafy patches. They mature earlier and reproduce sooner. Chimpanzees stay lower, must range farther for seasonal fruit, and travel more alone or in small bands when food is scarce. Those ecological differences mean that climate swings change group size, movement, and reproduction in ways that leave traces in bones and genes, and in where we find them on the landscape. In short, fragmentation of habitat and pulses of aridity can shrink populations, isolate groups, and send lineages down different paths, sometimes toward adaptation, sometimes toward local disappearance. Picture a drying riverbed and a scattered line of fruiting trees, apes moving quietly between those green islands. That broader pattern matters when we read human genomes for signs of a deep bottleneck. If our cousins in the trees show rises and falls tied to changing climate, then a dip inferred in human ancestry sits comfortably within a larger African biogeography of shrinking forests, shifting savannas, and scattered refuges. In plain language, a dip in genetic diversity can mean many small, isolated groups rather than a single sudden crash. The fossil and genetic record reminds us that apes once shared a common ancestry deep in the past, and that branching and fragmentation have been part of the story for millions of years. Was the story of our ancestors a single sharp collapse, or a patchwork of isolated groups weathering long seasons of stress and occasional shocks? The next question turns to those shocks themselves: volcanoes, rare catastrophes, and how they fit the longer climate story. When readers hear of ancient population crashes, their minds often jump to sudden dramatic scenes, ash falling like snow, skies gone dark, whole regions chilled and silent. That image is not wrong for some moments in Earth's story. The eruption of Mount Toba about 740 years ago is one such dramatic event in the record, a supereruption that sent vast volumes of ash into the air, and some have argued plunged summers into long, cold spells. Stanley Ambrose and others have proposed that such volcanic winters could have driven sharp population declines much later in human prehistory. But did a single eruption explain a genetic signal deep in the past? The timing does not match. Toba belongs to a far later chapter than the contraction some genetic studies place near 900 years ago. Around 930 to 813 years ago, the more grounded explanations point away from one-off cataclysms and toward long changes in climate and habitat. Evidence in the fossil and environmental record suggests that repeated cycles of drying, cooling, and landscape change could fragment populations, isolate groups in scattered refuges, and make gene pools smaller without a single dramatic disaster. Imagine a network of lakes and river corridors drying into ponds, each one becoming an island where small groups linger and slowly drift apart. Over many generations, These scattered pockets can leave a clear genetic signature without any single dramatic disaster. Fossils are sparse, and the genomic signals are model-dependent, so researchers read these clues with care. The key image is not a single night of ash, but many seasons of shrinking lakes, patchy forests, and thin corridors of green conditions that erode connections between groups and leave a genetic echo of small effective size. Next, we will look at how researchers pin ages on these events and test these ideas against the clocks in stone and bone. To tell time on the deep stage, researchers read rock and bone like an old ledger. Long sequences of sediment can record flips in the Earth's magnetic field and changes in chemistry. Paleomagnetism and isotope stratigraphy help line those sequences up across places. Where a clear reversal or chemical shift appears, it becomes an anchor that links a local site to a larger timeline. Teeth and bones carry other clocks. Uranium series and related approaches can date the mineral growth in a tooth, and electron trapping methods read the last time a grain of sediment saw sunlight. Each of these techniques measures a different process, so they work over different intervals and with different precision. In the field, a pale ribbon of ash or a dark buried soil can look ordinary until laboratory work turns it into a date. How do we know which layer is older? Those different clocks speak to one another. Volcanic ash layers give a radiometric anchor from which surrounding deposits gain. Age sediments reset their trapped electron signal. When exposed to light, tooth and bone chemistry record decay-based ages. When paleomagnetic stripes chemical stratigraphy, volcanic dates luminescence signals, and tooth-based clocks all point in the same direction. Confidence grows. Sometimes the clocks disagree, and that disagreement tells a story too. Reworking contamination or complex deposition are then investigated. A thin ash layer traced across regions can act like a ribbon tying sites into a single history and cross-checks tighten or loosen our sense of timing. That is how researchers situate deep events such as the contraction some argue occurred between the ages quoted in the record. No single method rules the story. Multiple independent clocks cross-check one another, tightening or loosening our sense of when climates shifted and people moved. Next, we will listen to what the sediments themselves remember: dust, pollen, and the slow pulse of lakes and seas. Beneath the slow push and pull of sea and lake sediments collect a quiet record. Each layer is a page where dust, pollen, and organic molecules settle and wait. Coarse dust can hint at distant drylands. Pollen tells which plants held the soil and water. Certain biomarkers, even molecules from human waste, can survive in those beds for centuries and millennia and signal past people on the landscape. At Horseshoe Lake, for example, lake cores preserved fecal stanols that rise and fall with human presence, a direct chemical whisper of population change. Read together these traces, sketch humidity, temperature, and the shifting mosaics of habitat that would have framed hominin lives: salt wind or freshwater reed, open grass, or thicket, a coastline that lengthens or withdraws with the sea. Researchers extract long, narrow cores like bookmarks, letting them read the sequence of those pages 1 centimeter at a time. A newer thread in this archive is environmental DNA, fragments shed by plants, animals, and microbes. Where preservation is kind, these traces promise to name species long gone from a site, adding texture to pollen and dust. But such molecular gifts are not uniformly available in many deep tropical sequences. They remain rare. When present, environmental DNA must be handled like a faint fingerprint, carefully, and with many checks against contamination. Still, when cores, pollen diagrams, and biomarkers agree, they give a steady cross-checked picture, a world of waxing and waning water, of abrupt aridity or gentle greening, and of places that could hold people or force them apart. Together, these archives let us trace where food, water, and shelter could be found. If sediments can tell us how the climate tightened, what then of the tools people carried into those changing spaces? That question is where climate records meet stone and bone. Even the simplest acts of sharing can hold a population together. When meat is divided at a fire, when a child is taught where to find tubers after the rains, when a group moves its camp to follow a river, knowledge is carried as surely as food. These social practices, learning by watching and doing, provisioning those temporarily weak and seasonal mobility along familiar routes, serve as buffers. They let local shortages be weathered without every group having to be self-sufficient. In such a world, a tool's shape or the memory of a spring matters as much as the tool itself. Skills and roots pass between people the way stories pass between nights. Skills travel in people's heads as well as their hands, so a community can recover knowledge after a bad year without relearning everything. But when the land tightens, those threads begin to thin. Pulses of aridity can shrink usable habitat into scattered refuges. Isolated river corridors shrinking lakeshores, coastal strips, so that groups meet less often. Reduced contact cuts both ways. It raises the chance that a loss of a key learner or a failed migration severs a chain of knowledge, and it also limits the flow of genes. Over generations, that isolation deepens signals in genomes that look like a small, long-term ancestral population. In people's bones and tools, we see hints of scattered lifeways. In the genes, we see echoes of isolation. What would a true near extinction leave in the DNA? Genomes preserve hints of these social rhythms, but they do not keep every detail. Interpreting those hints means cross-checking genomes with bones, tools, and the layered earth that held them. Only by joining those lines can we tell if the thinning was temporary or a passage through a narrow gate. Around the time deep in the Middle Pleistocene, when some models pick out a sharp dip, a contraction dated between 930 and 813 years ago, the world of our ancestors may have felt like a narrowing gate. Evidence suggests not a single cinematic extinction, but a patchwork of stresses, longer, harsher cycles of climate that turned broad habitats into islands of green. Lakes shrank to salt flats, rivers pulled inward, and the savanna, forest mosaic that once allowed for wide movement, could have broken into distant pockets. In those pockets, small groups clustered, a handful of families by a relict shore, a night watch around a low fire, a hand axe smoothed by use, and the palm that held it. Reduced connectivity across the landscape would limit mate exchange, and shared knowledge, making many local populations fragile even if some people survived elsewhere. One plausible pathway through the narrow gate is prolonged fragmentation. Many small, partly isolated refuges linked only by rare contacts. Another is a sharper contraction to a few richer refuges, river corridors, lake margins, and coasts that buffered food and water, followed by a slow, uneven rebound as climates eased. Genetic studies elsewhere have inferred founding numbers as low as a few hundred to a few thousand, which helps show how small effective sizes can be even without total collapse. Cultural tools like the Acheulean toolkit may have offered insurance, and social bonds like food sharing would have mattered as much as meat or tools. Yet these scenarios are not mutually exclusive, and models can confuse isolation with extinction. Or could the genetic whisper we hear come more from long separations than from a single catastrophic die-off? In this narrowness, ordinary daily skills could decide survival. When geneticists read a deep dip in diversity, one plain interpretation is a sudden collapse, a near extinction. But the same pattern can arise from quieter, more complex stories. Imagine a continent of many small groups each living by a river or on a lakeshore linked by occasional meetings and exchanges. If those links thin the genes that move between groups slow to a trickle, the overall signal in genomes then looks as if numbers fell everywhere at once even when many people still lived on the landscape. We also know that our picture is blurred by the scarcity of ancient genomes from large parts of the Old World. Without those direct windows, hidden contributions from now vanished neighbors can push models toward a single sharp collapse instead of a patchwork history. Put simply, genomes record not only how many people lived, but also how often they met. Selection and sampling add other mirrors. Strong selection on parts of the genome, a favorable mutation sweeping through a region, can erase diversity locally and make populations seem smaller in genetic reconstructions. Limited sampling of living diversity can overweight particular lineages, and archaeological gaps remind us that local disappearances are common. The Middle Eastern populations known from coastal caves, for example, fade from the record when climates shift. Could scattered declines, shifting contacts, and bouts of selection produce the same genomic shadow as a single catastrophic die-off? Quite possibly. Each of these scenarios implies different pasts, one of fragmented groups holding on in refuges, one of fluctuating bridges of gene flow, one where selection reshaped diversity without wiping out people. The next chapter considers the converse, the ways populations might rebound and spread again when the climate eased and old roots reopened. After the severe contraction between about 930 and 813 years ago, the deep record hints at slow loosening of the squeeze. As climates swung toward milder intervals after about 700 years ago, river corridors and lake margins would have come back to life. Reed beds whispering mud, soft underfoot shorelines offering fish and plants. In some places, the archaeological scatter thickened stone artifacts on North Sea beaches, and at coastal places suggest repeated visits, and by 500,000 years ago, a recognizable human presence is established in parts of northern Europe. Elsewhere, pockets of people persisted much earlier, with evidence of occupation in parts of Southeast Asia by around 900 years ago. These regional contrasts show that rebounds were uneven. Some landscapes opened like doors, others stayed fenced by drought or ice. Tools and habits carried across those doors, helping groups reconnect when the weather allowed. This reconnection was slow, replayed over generations, as people reused familiar routes and water holes. On a returning shore, reed stems might brush ankles while small fires hiss, and fish are cleaned, a routine that helped memory outlast one hard season. Such reconnections could produce the archaeological pattern. We see clusters of denser sites in favorable regions, broader ranges, where habitats linked up, and long stretches of silence where refuges remained isolated. Social networks shared knowledge of water and food, places, and mobility along familiar routes would have acted as glue, knitting scattered groups back together and spreading innovations. Because preservation varies, denser site clusters may reflect better survival of archaeological traces as much as larger past populations. Yet the record stays patchy, so historians of deep time must read these echoes with care. What traces would these rebounds leave in later bones and genomes as we look ahead to the next chapter? If a long stretch of our deep past passed through a time of very low effective population size, its effects would be subtle and cumulative rather than theatrical. Imagine bands huddled along shrinking river margins, the air dry, their stone tools clacking as someone sharpens a hand axe. Small scattered groups left fewer chances for genes to mix. In such a setting, chance plays a larger role. Random shifts in which variants survive and which vanish. Genetic drift and lineage sorting become stronger forces. Over generations, this can make some branches of the family tree disappear, while others become more common not because they were better, but because they happened to be in the right place at the right time. Books and studies have long proposed episodes when human numbers were thin, suggestions that ancestor populations sometimes fell to only a few thousand or to around 10,000 adults. Those are model-dependent ideas. If any part of the African metapopulation spent long periods small or isolated, the long-term consequence would be a patchwork of lineages, each shaped by luck and local history. This can help explain why later branches of Homo look different from one another, and why some genetic lineages seem so sparse in the record. It is important to keep this measured. A phase of small n does not script a single inevitable path toward modern humans. It only raises the odds that random change, local loss, and the survival of particular lineages mattered more. The echoes of such times could bias which traits and which genes survived into later populations, nudging the shape of our ancestry without forcing a destiny. What new samples and refined clocks might reveal about those nudges is the next question we must follow gently. If a headline says 99% wiped out 900 years ago, the calm question is not whether the models can say that, but what new data would make most researchers change their minds. Deeper, broader sampling of African genomes would be one clear test. Right now, genomes and mitochondrial studies give us hints, but more samples across different regions, and peoples would show whether a signal of extreme shrinkage is shared widely, or is instead a quirk of limited sampling. Better calibration of the molecular clock, the rate at which mutations accumulate, would also shift timelines. Small changes in that rate move inferred dates by large amounts, and tightening that clock would tell us whether the apparent dip sits where models place it or slides elsewhere. In plain terms, shifting the clock is like moving the labels on a family album. A small error can tuck a portrait into a different decade. Equally important are methods that do not treat size, structure, and selection as separate puzzles. Joint inference that models population size together with fragmented landscapes, and the tug of natural selection would help distinguish a true near extinction from a pattern caused by isolation or selective sweeps. Ancient proteins from tooth enamel and other durable tissues offer a hopeful avenue when DNA is gone. Refined protein trees could anchor fossil cousins and clarify branching order. Imagine a molar, its enamel still catching light in the field, yielding a protein signature that repositions a fossil on the family tree. This kind of find would nudge genetic interpretations. Such field finds have outsized power to change interpretations. What would convince us, in short, is more and better connections between genomes, molecules, fossils, and climate records. Then we can read the story more gently and accurately in the headlines that follow. Headlines that say, "wiped out," make a sharp picture one day, many the next day, almost gone. That image is easy to hold, but it often overstates what genetic and archaeological work can show. Some researchers note a severe contraction in our African ancestors sometime in the deep Middle Pleistocene, with dates given in one discussion as between 930 and 813 years ago. Other work points to bottlenecks at later times too. What the data actually record is a change in genetic diversity, or in the way lineages trace back, not a simple head count. Key assumptions: how fast mutations accumulate, the average age of parents, how populations mix, shift the timing and size of those inferred events. Small changes in those settings can turn a modest dip into a dramatic fall on a graph. Think of the result like a blurred photograph. Outlines are visible, but the picture's sharpness depends on the lens and light. It helps to read headlines like a map with many layers. Ask whether the claim rests on genetic models alone, or if fossils, tools, and climate records line up. Remember that population structure and long-term cultural movement can mimic abrupt crashes by lowering effective population size without a literal mass die-off. Sampling limits and natural selection on parts of the genome can also skew the picture. Little details from sediments, bones, or burned hearths can tip the balance between stories. Be curious about the claim and patient with the evidence. Science often offers several plausible stories rather than a single dramatic one. As we move next to why this deep time story matters, imagine refugees of a changing landscape finding shelter along a shrinking lake shore. What does that tell us about resilience? Next, we will look for where these different records converge. Today's crowded world can make the deep past feel like a different planet. Yet the record we have, faint bones, scattered stones, and the patterns written in our genes, hints that deep in the Middle Pleistocene, many researchers see a severe squeeze on ancient populations. Some argue that ancestors in Africa may have been reduced to on the order of 10,000 adults for a prolonged interval between roughly 930 and 813,000 years ago, a contraction that other accounts describe as lasting on the order of 20,000 years. Those are model-based readings offered with caution, but they paint a quiet scene: rivers shrinking, lakes retreating, patches of green and water becoming islands of life, where people gathered, traded knowledge, and shared what little the place could offer. In those places, survival would have depended on steady hands and steady ties. Night by night, small fires would have warmed bodies and bones. People would have watched one another while eating marrow and roots, guiding children and carrying the weak. Modest stone tools, learning past mouth to ear and roots to reliable water, would have been the true technologies that kept groups alive. From that long, narrow corridor of time, there is a simple lesson: variability is the norm, not the exception. The past does not read as a single catastrophe or a single triumph, but as cycles of narrowing and rebounding, of isolation and reconnection. This is not a story meant to alarm, it is one that asks humility. In short, the record requires careful interpretation, not alarm. If we want to decide between a population collapse and a tale of fragmented but persistent groups, what kinds of evidence would move us? And how might we find those traces next? We live among many millions now, and yet deep in the Middle Pleistocene, the record suggests a thin, fragile world. Some genetic and archaeological readings point to a dramatic contraction of our ancestors between the span around 930 and 813 years ago. Other scholars picture a less dire scene. One reconstruction even frames a later small population measured in thousands of adults rather than absolute disappearance. Both views rest on models: sparse bones and scattered sites. Stone keeps its own memory, a hand axe, its edge worn smooth by use, tells of work done and food shared. Lake margins and rivers that once fed small bands remind us how water and forage shaped movement. Bones broken and buried in the earth hold traces of lives that were not always easy, but were lived with steadiness and care. A modeled fall in effective population size can mean fewer breeding connections or isolated groups, not that every person vanished. Our story is built slowly by many methods and minds, and it changes as new pieces arrive. Some researchers stress sharp drops in genetic diversity. Others point out how population structure or shifting connections between groups could look like a collapse in the data. This is not a tale of doom, but of limits and resilience, of groups finding refuges of tools and knowledge carried from hand to hand of slow recoveries when climates eased. There is humility in the work we reconstruct from fragments and probabilities, not scripts. In the quiet light of those old camps beneath the patient witness of stone and bone, we are reminded that our shared past is complex and alive to new readings. We keep asking with the care of slow lanterns testing and adjusting as new fragments arrive. What will these slow records teach us next?